Okay. Funciona? Huh? Okay. Okay. Yeah, it's great to be here. Thanks for the opportunity to talk about something that has actually occupied my mind for quite a while. And I want to put that in maybe a larger framework. The framework as doubts as a scientist. And I think it's doubts in terms of the terms we use, the appropriateness. Well, we were just talking about how do you actually think about genoma, so what is a species? And on the other hand, also like doubts about all the statement that you often read in books, in textbooks, and the validity. So often you have statements that are not really based on a lot of facts. They sound good, they may be intuitively like appealing, but they may still be wrong. And so it can go to the very heart of a person who's been working with epiphytes for a while. So what is an epiphyte? It's seemingly easy because an epiphyte is a plant that is growing non-parasitically on another plant. So is that an epiphyte? That's Nephidium crassifolium. And I think the species is still an epiphyte. Of course, the individual is not. If you just look up in, that is actually a picture I took here close to San Jose, where you have, that's a Catopsis nutans, which is an epiphyte. Not really in that situation, but I still think, well, there's some leeway. This is not a monocot. That's a papaya. But that papaya is epiphytic, is about 1.5 meters tall. I just took that photograph a week ago in Gamboa, Panama. And this is a royal palm, like 3 meters tall in a Guayanacaste. And well, typically Roystonea is not really an epiphyte. And the neatest picture is actually this. That's in 15 meters height. That's an agave, an epiphytic agave. And of course, you can now start in like having this anecdotally, but I did this in a more analytical formal way by just looking at information in the literature where you can look at the description of species, and it says epiphytic, rarely terrestrial, or it says litophytic and terrestrial. And we did this for 1,500 species of ferns and collected 6,000 observations, individual observations. And so on average, 4 observations per species, and we then had an average per species, and we put that in a triangle. And you may be familiar with the famous Grime triangle or the Silvertown triangle, and now I try to have something like a Zotz triangle. Here you go. So what you see here is that a lot of the species in Elaphoglossum are really mainly epiphytes or exclusively epiphytes. And if you go to the upper corner, all those are like almost exclusively litophytic species. And on the right side, then you have something that is exclusively terrestrial. But you see that a lot of these dots are somewhere in between that space. So they're not, if you think of like, what is an epiphyte? Is this an epiphyte? Or is this an epiphyte still? So we're running into the problem that these categories are actually put on top of something that is actually a continuum. So there's more like a preference. So of course that for me as an epiphyte person is kind of difficult like to deal with. And I will still like go back in the presentation now that I talk of epiphytes, but always please with the idea that maybe I'm not so quite sure what I'm talking about. So the doubts about things that we have statements on what is something. So the velamen radicum is nicely described 200 years ago by Link as something that is a uni or multiseriate rhizodermis that consists of dead cells at maturity. And important that you have these PCs, which is not a computer, but which is a passage cell. So of course, functionally, it's important that something that is in the dead tissue, nutrients, water, that can pass into the living part of the root. And that's accomplished via these passage cells. So that is something that does exist, no doubt. But is what we read in the textbooks, is that actually where we find it, how often we find it, is that correct? So there's quite a consensus in the textbooks. Like here, in the arrow roots of some epiphytic orchids and aroids, the multi-layered epidermis develops into a dead velamen. Similar statements here. So And a velamen you find in epiphytes, whatever that is, and you find it in aroids and orchids. And I'm not only bitching about others, so that's a paper that I wrote 20 years ago. So where I also said, like, it's an unusual morphological structure that you often find in epiphytes. Well, it's vague enough that I can say I did not claim that it's only found in epiphytes. So, what's the evidence? If you go to the orchids, then yes, there's a lot of evidence that you have, in orchids, you do have a velamen in many, many, many species and thousands of species. And you have at least looked at it in hundreds of species, so I think we can generalize. But if you do the same thing for aroids, then there's some information for Anthurium, exactly 13 data points out of more than 1,000 species. So I don't think that's a convincing evidence that you can make the claim that it's really found in Anthurium, let alone in aroids. And so like in Thomas and Monica Carlson's nice phylogenetic analysis of the genus Anthurium, they don't even use the velamen. Because, well, you need enough information that you can include it as a trait. So I think, well, it's not that clear. And so how typical is it? And so, well, just do it. So we did that study. We looked at 80 Anthurium species along the whole tree that we could use from Tom's work. And here you see for 2 species that you have that type of a tissue. But here's the story. It's as common in the terrestrial as in the epiphytic taxa. So yes, it is common in Anthurium, but it has nothing to do with epiphytism. And I have to show a tree, of course, as well. So here's a tree. And so all these terrestrial things, those are the terrestrial ones that do have a velamen. So it's scattered all over, and you could come up with the idea, maybe those are just like epiphytes that have fallen to the ground and just kept the velamen because it's not that much of a disadvantage. But, you know, I published something that a while ago where we looked at the occurrence or the reports of recurrence of velamen in all around the monocots, and you see that it's found in all kinds of things. So in 74 non-orchid genera, in families where we have no epiphytes whatsoever. So the argument that it's just like a fallen epiphyte is not really valid, at least not as a general one. So I think claim rejected. We could have known if, as Tom told me, if he can read German. Because then he could have read what Karl Goebel wrote in 1921. He said, das Vorhandensein eines mehrschichtigen Velamens ist nicht, or maybe I help you. So this is the English version. The typical textbook description of the velamen radicum as a structure found exclusively in epiphytic orchids and aroids is wrong. That's a simple statement. And that was known 100 years ago, but forgotten in textbooks. Number 2, function. So is that clearer? There's a wonderful review by Pridgen, 1987, where he looks at the description of the velamen, and he states these main functions. So the capture, storage of water nutrients, reduction of transpiration and water loss, mechanical protection of the living cortex, reduction of the heat load on roots growing in exposed conditions, and one more that came up later. The problem with this is that there was very little evidence that, well, it makes sense, but experimental evidence that is backing up the claim of Pridgen. So, well, then you have to do it. And that's what we did, now more than 10 years ago, that we looked at one aspect of the first one, capture, storage of water nutrients, more specifically using a suggestion by Wendt, where he said the primary function of the velamen is actually to capture the first loads of water, which are richest in nutrients. So demobilize them so that then you have time to take them up. And the experiment that we did was with rubidium, which is an analog of potassium, and actually radioactive in that case, so that we could look at different treatments, velamen intact, velamen dead, or velamen removed. And you see in all cases, the uptake, the seeming uptake, which is mostly adsorption, was identical. But if you then put them in distilled water and look what's happening, you see that the one treatment where the velamen was removed lost it all. So it's not staying inside. Whereas the other two treatments are more or less like really like very effectively just capturing this so that the plant has all the time it needs later to take up those nutrients. If you do the same thing with something that is not charged, so a sugar, then it doesn't work. Then it's just getting lost. So it's really charges in the cell walls that apparently like demobilize it. So, although Pridgeon didn't really know, well, he was right. The second, reduction of transpiration water loss, that we did in the same paper, and yes, it does work. The third one is kind of hard to, well, it makes sense, and therefore it gets a tick, although I don't have any evidence. But I think it makes sense. And now the last one, which also makes sense because, well, you have that white velamen when it's dry. So, but is it true? And we did that experiment with infrared. And on the right side, you see where we measured temperature just at the exodermis, where you have the living tissue, and at the surface of the velamen. And you see that that's the time scale over like 30 minutes, increase, and then a more or less stable temperature at a IR load that is typical to a tropical climate, lowland climate. So you see it really doesn't help much. It helps a little bit, but not much. So I would, but that's 8 species only that we were included. So I would still leave that open. So do another study. So different people said just maybe some details that are different. Maybe there is something on it because it was kind of difficult to explain why these things can exist at all, because we also looked at temperature tolerance, and they're really getting hard to temperature tolerance limits. So I can hardly explain why they exist, where they exist with this. And then the last one, UV protection, that was nice because Chumicki et al., they did not only propose something, but they actually tested it. And they showed that, yes, these things do produce flavonoids, and that is induced, and that helps them really as a UV protection measurement. So, a number of ticks, but there's more. Like if you look finer, you have at the, it's kind of, don't we have a point or something? Eso? ¿Dónde? And there's another button. Okay, the green one. Okay, so here's the patches cells. And so often, like you have that in species that you have these so-called tylosomes, which are just exclusions. And it stands to reason again that this has something to do with the control of the uptake. Maybe also like acting like a valve, so that it's just what goes in is possible, but then it doesn't go out. But that's pretty much unresolved still. So that is certainly something that would be interesting to look at. And what you just did in Anthurium, we looked in detail at the structure of this velamen. And what you can see here is that you have all these holes or slits. And of course, you have a lot of variety, but whether that's functionally relevant or not is really unexplored. But that's really, if you are into mosses maybe, or that's a monocot conference, but still, well, if you think of the genus Sphagnum, for example, they do the same thing. So, but again, you have form, but it's not clear whether that's functionally relevant. And that's actually on the agenda in my lab in the next couple of years. There's some theoretical background by this paper by Hauber et al., but they looked at the function in terms of water uptake, water storage, and water loss at the different levels. So how many cells you have and how these cells are connected with and without holes. And of course, like the function in area roots is that they can also at least potentially photosynthesize, although there's not much data on that, how general it is. And so I have a student of mine who does his bachelor thesis, Pepe Goshin. And so what you have the maximum and the minimum values of electron transport rates of roots and of the leaves of the same species. And you see that you more or less have the same range, which was kind of surprising, that you really have very high rates of, it's not CO2 uptake, it's photosynthetic activity, whatever that means. Because of course that is in competition if you have a wet velamen in terms of CO2 uptake, because that is not really good for CO2 uptake. So that's in leafy ones, but it's much more of a thing. So transmission And the velamen thickness is something, again, a functional thing, because if you have a velamen that, of course, also absorbs some of the light and may not be conducive to do a lot of photosynthesis, but here you see that a relative simple correlation that the thicker the velamen, the more light is absorbed. But still, it reaches 70% or 95% in the extreme values, which if you are exposed in a tropical tree crown, I think is still more than enough light that you need to run your photosynthetic apparatus. So that does work. But of course, it gets more complicated in those orchids that do not have leaves, because then the roots have to do all the job. Then it's not just like reducing your carbon expenditure, but you have to have a positive carbon balance. And so in that case, you have to deal with that problem that if you are wet, then you have a problem with actually the uptake of CO2. And they have this so-called pneumatodes. So those are areas that can't be wetted. So that allows CO2 actually to penetrate to the living part of the root and allow photosynthesis. But again, for example, the role that CAM plays in all this, that's also, again, something that is not really explored. So the take-home message that I want to give you, if someone is interested not only in phylogenetic trees, but maybe also in some function ecology. So here would be something to do, because there's a lot of things, open questions, and since I really believe in diversity, and so we don't have our Arabidopsis. We can't have just, this is the orchid I want to study, but its diversity is real. And so you really have to do a lot of like different species to come up with generalizations. And that's, of course, just jumping back to the initial comments, a problem we often have, that we have one or two studies and then we already start generalizing. And since diversity is real, and so the variability is not just noise, but it's real, it's really important to do this. And so I would be more than happy not to compete with you, but not even collaborate, but just like having other people who I may interest in doing interesting studies with the velamen. And on the one hand, of course, with these aerial roots. But I think as interesting as it is to work with aerial roots with a velamen is those terrestrial ones. So what is the function in the terrestrial environment? It seemingly made sense from the start that all these aerial things do have a velamen. But if many, many species on the ground have a velamen as well, so what are the consequences there? And that is not at all clear. And I invite you all, well, if you have someone to interest in working in this. I want to say thanks to a lot of collaborators. For example, these trees are not my trees. So those, I'm completely ignorant of that. They produce those, like Dirk Albach. And of course, money is an issue, you all know. And thanks to all of you for like attending this. And if you have questions, here I am.
Thank you. Time for questions.
Thank you for your talk. My question is about the tylosomes. I studied the velamen structure in terrestrial orchids, the spiranthoid orchids, and we found not only that there is velamen, there are tylosomes, but there is also variation in the structure of the tylosomes. Mm-hmm. There can be plates, ridges, or rods. Yeah. Do you have any ideas of what this variation means?
Well, it's the same as with the holes and the slits and all this, where that's just like variation that is functionally, well, the same, or whether it's different. I don't know. I really, yeah.
Any other?
So you mentioned in the terrestrial species, is there anything in common in the terrestrial species? Like I noticed that there was velamen in Cyperaceae. Are they semi-aquatic, or is there some ecological correlation that you can imagine between?
Well, it's more like in Mediterranean semi-dry environments, and Benzing actually proposed the hypothesis that that this allows in a often like going through these shrinking processes that would maybe tear apart a root that is kind of glued to the soil particles that move. And that would allow them to keep a certain distance without having a physical rupture of the whole thing.
So soils that dry out.
Yeah. It's worse because in some cases it's not even clear whether what people call a velamen is a velamen. So there's this a velamen in the bromeliads, which has been proposed by some Brazilians, and then another Brazilian actually, he did a very detailed study to show that the ontogeny is different and actually the function is different. And then you shouldn't probably not call it a velamen because I think it's the whole package. Just having like a dead rhizodermis is not enough to be a velamen probably. This Tom? I don't know.
So another possible function of velamen in cyclically dehydrated soils is simply decoupling the roots hydraulically from the dry soil. The large pores, relatively speaking, in the velamen are going to drain more rapidly than the pores in the adjacent soil, and that prevents the water loss into the soil from the live roots, so just as the velamen in aerial roots prevent the water loss to the atmosphere after a rainstorm.
Yeah, it would be. It's 0.2 of Pridgeon's functions. Yeah. So it's always like, does it hurt or is it really functionally relevant? Yeah.
One more? Yeah.
I wanted to ask you if you've studied Cyperaceae, just because they're so interesting. They can start as terrestrial and then climb and become, so they're hemi-epiphytes. Many of them are associated with streams.
Yeah, well, you open a box if you talk with me about so-called secondary hemiphytes. It's I don't know whether they do have a velamen. And the other thing in terms of whatever is what I would call climber, whether that's in philodendron or rhodosparta or like Cyperaceae, I still have to be convinced that there's anything that becomes an epiphyte. And it's not just a climber. And it's just like when I look out of the office window in Oldenburg, I see an ivy. And an ivy is just a liana. And I don't know why we have to call a philodendron or a Cyperaceae not a climber. So, but so that's a liana. That's a definition of a liana. Or if it's a vine, if it's a herbaceous thing. So. But when I talked about aroids and the velamen, I'm not aware of any study that has looked whether the few things that I did, like looking at a philodendron or a monstera, they don't have a velamen. So I don't know whether, apart from Anthurium, I don't know. I don't think there is a velamen anywhere besides in Anthurium. But there it's pretty common. But if you looked at the tree, like the basal parts, did not have a velamen. They're actually, coincidentally, Glidimyoides and Flexili, they are what I would call climbers. And they don't have a velamen.
Thank you.